Hey, Mr. P here. In this video, we're going to talk specifically about the ultrastructure of cells, which means we're going to dive into that cell membrane and we're going to talk about what the cells are actually composed of. We're going to highlight a few differences between prokaryotic and eukaryotic cells. This is kind of a follow-up video to the cell theory videos that we've done in the past. And so we're going to try to move through the ultrastructure of cells or the anatomy of cells rather quickly. A lot of this is probably review for you. And so hang on, make sure you're taking notes. Here we go. So the first thing we got to do before talking about the anatomy of cells is we have to look at how the cellular anatomy is actually studied. The main way to study cellular anatomy is with the use of microscopy. So microscopes can vary in size, they can vary in energy source, they can vary in structure, but the two types of microscopy techniques that I would like to highlight is with the use of a light microscope and an electron microscope. So obviously this picture up here is an electron microscope. You can tell that it is an electron microscope other than just the fact that it says electron microscope because it utilizes computers and monitors to help display what the microscope is actually viewing. The way in which an electron microscope works, which is quite a bit different than the way light microscopes work, is that electron microscopy utilizes a beam of electrons, which are the actual electrons when we think about subatomic particles, to bounce off or through an image in order to put together three-dimensional either surface kind of structure image or the way in which the image looks in more of a transverse way. So, light microscopes, we'll start there. These are the actual microscopes we use in class. Okay, light microscopes utilize a light source as an energy source. You're going to focus that light through a series of lenses and through the sample in order to magnify the image that can be either alive or dead, meaning it can be alive or preserved, both of which can be observed with light microscopy. The difference between the electron microscopy techniques in that transmission electron microscopy, which is often just shortened to TEM, transmission electron microscopy, and scanning electron microscopy, SEM, is that they both utilize electrons as the energy source. They're both going to use a series of condenser lenses, but the difference is whether or not you're looking at the surface or looking through an image, okay, or looking through a specimen. In a scanning electron microscope, which is exactly what it looks like, we are scanning the surface. So we can actually get a pretty detailed three-dimensional look at the surface detail of a specimen by shooting a beam of electrons off of this preserved specimen and then collecting all of those bouncing electrons with a detector, which gives us surface detail. Okay, so just like scanning a document, which means we're just scanning to look at the surface of the document, we're going to utilize the beam of electrons and detect them as they bounce off. In a transmission electron microscopy, we are actually going to send those electrons through the sample, and that's not going to give us surface detail like we can see here. It's going to give us kind of that X-ray type image where we can see internal structures of a cell, both of which are really important, but both of which have a very kind of specific purpose. Light microscopy, we can only look through the surface, meaning we can't actually get surface detail, but is a very low-cost option when we're studying cells. So light microscopy, very low cost. Transmission and scanning electron microscopy, very expensive, but can also resolve a lot, has a much higher resolving power, right? We can look at images that are much smaller when we use electron microscopy than we can with light microscopy, and it's all about resolving power of the microscope. Notice that the resolving or resolution limit of a light microscope is 200 nanometers, which means 200 nanometers is kind of the smallest distance we can actually resolve successfully with a light microscope, whereas with an electron microscope we can get that resolution down to 1 nanometer. So this is basically 200 times better. And so what do I mean by resolution or resolving power? Basically, if there are two dots very closely packed, resolution is kind of the ability to distinguish between those two dots. If those dots are 1 nanometer apart, okay, and that's 1 nanometer, with an electron microscope, we can actually resolve and see that those are two specific kind of standalone points. Whereas with a light microscope, if these were 1 nanometer, they would be so small that they would still appear to be one point, if we could even see that point at all, right? So, very, very important, but light microscopes are much more cost effective, right? It's what schools have. Some of your upper universities have electron microscopes, but not typically a microscope that younger students would have access to, right? So, light microscopes allow us to see the structure of a cell, but an electron microscope allows us to see the ultrastructure of a cell. Structure of a cell would be kind of those larger structures that are more pronounced, that will be accessible and observable with light microscopy. But to see those ultrastructures, you have to use electron microscopy techniques. And as we get into this lecture, and as we get further into the lecture, you're going to see that I'm going to actually show you actual electron micrographs, and then we're going to try to deduce function of that cell based on structure. And one way to deduce the structure and function of a cell is that you have to be able to resolve and look at the ultrastructures of the cell in order to kind of pick up clues as to what the cell is likely to be used for. Okay, so cellular structure via light microscopy. Notice that these are all light microscopy or light microscope images. Notice that when you look at this particular image, you see individual plant cells. You know they're plant cells because they have these green discs called chloroplasts, right? Those are the organelles that are associated with photosynthesis. But notice we can't see into the chloroplast. We can't see individual smaller organelles. We can only really see those chloroplasts, which are the largest or one of the largest organelles within that cell. And we can't really make out much detail within any of those ultrastructures of the cell. Here we get into actual plant xylem and phloem tissue. We can't really see cellular ultrastructures. Again, we can see kind of these big cell outlines, but in order to really dive into any one of those particular cells, we'd have to use an electron microscope. Here we can also see cells. We can see nuclei, but we can't see individual organelles associated with those cells as well. So we can observe the cells, we can see the nuclei, we can conclude that they are in fact eukaryotic cells, but we can't see ultrastructures of cells. And then here, this is another plant cell. We can tell because of the really thick, robust cell wall. But again, other than the nucleus, we can't really make out any of those ultrastructures within the cell either. So we can get good images with light microscopy, but we can't get great images with light microscopy. So if you contrast that with electron microscopy, notice that when we dive into individual organelles, we can see the ultrastructures of those organelles in much more detail. So this is a mitochondria. You can see individual cristae, the folds of that inner membrane. You can see individual ribosomes, these bumps along these folded membranes, that is the rough ER. Right? Into this plant cell, we can see that these are the chloroplasts, and unlike the light microscopy picture in the previous slide, we can actually see individual discs within those chloroplasts. So we can see the individual thylakoids, we can see the individual grana, we can actually see kind of into that nucleus, we can see the nucleolus, we can see vacuoles and vesicles, okay? So we can see a lot closer, and we can make out much more detail associated with these cells. Here we can see the Golgi apparatus. We can see the actual separation of all of these membranes. We can see the vesicles and vacuoles that are leaving the Golgi on that kind of transverse side. And then here is a very clear image that we can see not only the interior of the nucleus, but we can also see all of the rough ER and the endoplasmic reticulum, as well as the ribosomes that are attached to there as well. So again, hopefully between the last two slides, you can see that light microscopy and electron microscopy both have specific places within biology, depending on what you want to use them for. Okay, light microscopy, obviously a very low-cost way to get good images in order to kind of see holistically structures of the cell. But if you want to dive in and see the ultrastructures, again, there's a difference between structure and ultrastructure. Light microscopy gives us structure. Electron microscopy gives us ultrastructure. So, Let's dive into what prokaryotic and eukaryotic means. Remember, prokaryotic cells are cells that are pre-nucleus in structure. They do not have a nucleus. In fact, they are much smaller and simpler all around than the eukaryotic cells. An example of a prokaryotic cell would be bacteria. Prokaryotic cells only have very specific simplified structures. The structures that will typically always be present within a bacteria prokaryotic cell would be the cell wall. You can see the cell wall here on the outside of the cell. It's this kind of darker orange structure that is right outside of this green cell membrane. This particular prokaryote has a slime capsule called a glycocalyx on the outside of that cell wall, so it actually looks like there's two cell walls on this particular bacteria, but notice that the peptidoglycan-based cell wall is this interior kind of darker orange or tan cell wall. That is right on the outside of this cell membrane. Plasma membrane, all cells have a membrane. It's basically what regulates entry of materials into and out of the cell. It also serves as a boundary between inside and outside of the cell. I mean, a cell that cannot regulate materials coming into and out of the cell or keeping a boundary between what is inside and outside really isn't a cell at all. So you have a plasma membrane, which in this particular picture is green. You have a cell wall, which is just outside of the cell membrane and is always thicker than the cell membrane. And then in some bacteria, you have also a glycocalyx or a slime capsule, which just helps to protect the cell even further than the cell wall. Flagella can be present or doesn't have to be present. Flagella can also be singular or multiple, but the flagella, which are these pink kind of purpley structures, are all about cell movement. So those are the tails that are made of microfilaments or microtubules that are going to kind of twist in its environment and propel the cell in the directions it wants to go. Pili are these hair-like projections on the outside of the cell membrane. They extend through the cell wall and the slime capsule if there is one, but they're all about cell adhesion and conjugation. So most of the time, pili are all cell adhesion. These are the things that actually kind of stick the bacteria or the prokaryotic cell to its environment. But there is typically one pili that is kind of more complex and is called the sex pili. It is, or the conjugation pilus, but that is the particular pili that can actually take the bacteria, sometimes in the form of the plasmid or the bacteria's DNA, and can actually inject or transfer DNA from one prokaryotic cell to another prokaryotic cell. It's similar to sexual reproduction, but remember, prokaryotes always go through asexual reproduction. It's just a way for transformation to take place, right? If bacteria cells have to swap DNA in some ways, bacterial resistance, or even going to kind of come up when we talk evolution, conjugation is really important to bacteria, and conjugation is actually achieved through the use of a sex pilus. So ribosomes are those structures in any cell that produce protein. So they are the site of protein synthesis. In prokaryotic cells, they go through protein synthesis utilizing these 70S ribosomes, whereas eukaryotic cells are going to be 80S ribosomes. Nucleoid region. The nucleoid region is essentially this kind of nucleus-like area. It is not a nucleus because it is not incorporated or does not incorporate a membrane around the DNA. But the DNA is kept kind of tightly condensed or tightly coiled into a region. It's just not a nucleus because it is a prokaryotic cell, so it is called a nucleoid region. It is all about DNA storage. Plasmid is kind of an accessory DNA. It is a smaller ring of DNA. It is specific to prokaryotes. A lot of times when you get into bacterial resistance, the plasmid is what is actually in charge of that bacterial resistance. Cytoplasm is just the structure that kind of is liquid, aqueous, that all of the ribosomes, DNA, plasmids are contained in. So it's kind of the jelly-like substance that fills the membrane to give the bacterial cell substance, right? So we've got cell wall, plasma membrane, flagella, pili, ribosomes, nucleoid region, plasmid, and cytoplasm. All are structures present within prokaryotic cells. Prokaryotic cells are going to divide by binary fission. This is a different process than eukaryotic cells go through. Eukaryotic cells are going to divide via mitosis, which is different, and mitosis is going to be covered in a different topic. But binary fission is essentially duplicating, it's basically two steps, right? We have to duplicate the DNA. So one cell is going to typically have one bunch of DNA, okay, one molecule of DNA in the nucleoid region. That one copy of DNA has to duplicate into a second copy of DNA. It is identical to the first. Once you copy the DNA, you simply move this copy to one side, this copy to another side, and then you start elongating the cell. Okay, so the cell starts elongating, it pushes out, pushes out, pushes out. As the cell elongates, it pinches in the middle until eventually the pinching in completely separates the cell into two. Okay, these are identical daughter cells to the original parent cell. And this is a form of asexual reproduction. So what happened? Just duplicate DNA, push the DNA to opposite poles of the cell, pinch in until you completely separate the cells. Now we have two identical daughter cells that are identical to the original parent cell. That is binary fission. Very simple. It's basically cellular cloning. Okay? How can bacteria cells replicate so quickly? It's because binary fission doesn't take a long time. I mean, a lot of bacteria can actually replicate every 20 minutes. So not only are they duplicating themselves within the 20 minutes, but they're actually living out the whole life cycle of that cell. So one cell becomes 2 in 20 minutes, 2 becomes 4 in 20 minutes, 4 becomes 8 in 20 minutes, and so you can see how quickly a colony, or a pathogenic colony of bacteria can actually establish itself in a relatively short amount of time. So let's contrast that with eukaryotic cells. Here you can see an animal cell, you can see a plant cell. Both animal and plant cells are eukaryotic. You can tell that they're eukaryotic, one, because they both have a nucleus. Okay, eukaryotes have to have a nucleus. They have to contain the DNA within the nucleus. They contain the DNA within a core called the nucleolus. There is a noticeable membrane around that nucleus, and there are a variety of organelles that are either associated with membranes themselves, specifically the endomembrane organelles, or they are not associated with membranes like the ribosomes, but the ribosomes are larger, and we're going to go into kind of what ribosomes do specifically later in the presentation. But the use of membranes allows for compartmentalization, which is really, really important. We've already talked structure and function of compartmentalization. You know the importance of surface area to volume ratios. We know that by limiting volume, we are actually increasing our surface area to volume ratio. And so we, by utilizing membranes within a eukaryotic cell, can actually create compartments that further increase surface area to volume ratio and can give rise to not only increased efficiencies, but larger and more complex cell structures as a result. Okay, so the more efficient the cell is, the larger the surface area to volume ratio is, the more compartmentalization can take place, and the more complexity can result, or the increased compartmentalization leads to an increased specialization, so to speak. These eukaryotic cells are the base for all organisms that belong to the groups of algae, protists, fungi, plants, and animals. So everything on the planet that is multicellular is made up of eukaryotic cells. Eukaryotic cells lead to compartmentalization, which leads to cell specialization, which leads to cell differentiation, which leads to multicellularity. Okay, it's really, really important. So, we're going to go through all of these organelles, the structure and function of these organelles. We're going to try to go through them quickly. Again, I think a lot of these organelles have been instructed on in the past. I think that there are probably reviews to you by now. But organelles associated with all of these eukaryotic cells would be things like the nucleus, endoplasmic reticulum in two forms. You have rough endoplasmic reticulum and smooth ER, which we typically just say smooth ER and rough ER. Okay, both endoplasmic reticulums. Ribosomes are the ADS ribosomes associated with eukaryotic cells. Golgi Apparatus, lysosomes, mitochondria, chloroplasts, vacuoles, centrioles, and cytoplasm all belong to the class of cells that are eukaryotes. So, if we start in the nucleus, we know that the nucleus is a structure that contains the DNA within eukaryotic cells. The DNA is kept within the center of the nucleus. Notice the nucleus has this kind of dense core called the nucleolus. Okay, and the nucleolus contains the DNA. The DNA is very densely packed within the nucleolus. Okay. In eukaryotic cells, the chromosomes are linear, and what that means is that the chromosomes are actually X-shaped. In prokaryotic cells, the chromosomes are actually all folded up, but they're actually folded up large rings of DNA. So the chromosome is not an X-shaped like it is in eukaryotic cells. In prokaryotes, it's actually a large ring. Okay, so that's what we say. It is circular in nature in prokaryotes and linear in nature in eukaryotes. The nucleolus is where RNA is made, specifically mRNA. mRNA is going to be made in the nucleolus and is actually going to travel out through these nuclear pores. And so later in the IB syllabus, we're going to start talking about protein synthesis and RNA transcription. Those RNA molecules that we're going to talk about in detail are made in the nucleolus, which is why I'm talking about it now. The nuclear membrane, or the nuclear envelope, is composed of a double membrane. It is porous, and that only allows for increased functioning, right? So we've already said that DNA is kept within the nucleolus. It is so large that it will not be allowed to come out of the pore, but the mRNA that is made during RNA transcription is going to be made using the DNA as a template, but it's going to be made in the nucleus and is going to be allowed to leave through those nuclear pores. Our RNA, or the ribosomal RNA, is actually going to be made in the nucleus as well. And so mRNA molecules are coming out through the pores, but also the ribosomes are going to be made in the nucleus, and then they're going to be allowed to come out through the pores as well. So we have to be able to maintain a boundary between the inside of the cell and the inside of the nucleus, which is what this double membrane does, but because it's porous, it increases or allows for compartmentalization of the DNA, but actually allows for RNA processing to take place as well. And like I've talked in previous videos, most cells are going to have a single nucleus, but some cells can actually have many nuclei, like in the exceptions to cell theory video, we talked about the muscle fibers, right? They have nuclei all the way along the surface of the cell. But DNA, the DNA kept within this nucleus in non-dividing times is going to be in the form of chromatin, which is uncondensed, non-dividing form. It is this long kind of fiber form of DNA that is associated with histones, which are the kind of the proteins that are going to be used as spools to keep our DNA structured and untangled. In dividing times, during cell division, during mitosis, meiosis, these chromatin fibers are going to condense into structures called chromosomes, which are these X-shaped linear chromosomes in eukaryotic cells that are going to become condensed, both of these DNA forms are going to be kept within the nucleus and important for their particular time within the cell cycle of that cell. So if we focus on the DNA, for instance, again, eukaryotes, okay, eukaryotes, cells with the nucleus, are going to have their DNA in these linear chromosome forms, and these are only going to be achieved by supercoiling this chromatin fiber, which is further condensed using these histone proteins together in these kind of octamers. Notice we have 8 of these histone proteins, and each of those 8 octamers are going to be wrapped twice. And there's going to be future videos on nucleosomes, there's going to be future videos on these histone proteins specifically, but I just kind of wanted to introduce these structures because they are in fact kept within the nucleus. And it's important to talk about them when we talk about the nucleus so that, again, you can understand structure and function of the nucleus. We have to keep the DNA safe. We have to keep the DNA away from the enzymes and the lysosomes associated with the cytoplasm. One way in which we do that is through the use of that nuclear membranes, okay? But we make things in the nucleus like the mRNA, rRNA, and those ribosomes and mRNA have to be able to get out so that they can do their job within the cell cytoplasm. So, ER, Endoplasmic Reticulum. Again, like I said, there is smooth ER and there is rough ER. There are two specific ERs because there are two specific functions associated with the ER. Endoplasmic reticulum on the rough side, which is where I'm going to start, is an extensive system of flattened membranes. Notice that this is kind of the purple side. The rough ER in this diagram is this purple kind of interconnected flattened sacs that are associated with ribosomes. So the reason it is called rough ER is because when you look at it under a microscope, it appears to be bumpy, and those bumps or those studs are actually the ADS ribosomes that are attached, because the purpose or the function of this rough ER is to utilize those ADS ribosomes to synthesize proteins, transport in vesicles those protein products to the Golgi apparatus so that protein modification and maturation can take place. The rough ER is always associated closely, or is more closely associated with the nucleus, which is why the rough endoplasmic reticulum, when viewed under a microscope, will typically always be adjacent to or right outside the nucleus. When you go out from the nucleus and out past this rough ER, you're going to get to a system of kind of these tubules. The structure of the smooth ER is going to look different than the structure of the rough ER, but that's because they don't have the same function. Structure dictates function, and one of the major structural differences between the smooth ER and the rough ER is that the smooth ER is not associated with ribosomes because the structure doesn't need to be, or the function doesn't need to be associated with the ribosomes, because the smooth ER is going to be utilized in the synthesis of other things. The ribosomes that are associated with the rough ER are going to be used to make proteins. The lack of ribosomes is going to be illustrated in that the smooth ER is going to be utilized for a variety of other reasons. So we're not going to be producing proteins like we did with rough ER. We're going to be producing lipids. We're going to be producing sex hormones like testosterone and estrogen. The smooth ER is often in charge of detoxification of drugs in the liver. So liver cells specifically are going to have a large proportion of smooth ER to rough ER because the liver is all about detoxification of the blood. And so that cell is perfectly structured that allows it to function in this way. Storage of calcium ions in muscle cells, and those calcium ions are needed for muscle contraction. Muscle cells have to have a large proportion of the cell devoted to the smooth ER, because if you don't store enough calcium ions, you can't properly contract the muscle. And a muscle that can't contract is obviously going to be a problem. And the smooth ER is going to help the liver release glucose into the bloodstream when blood glucose levels get low. So, two different divisions. Notice that half of the ER is rough and half of the ER is smooth. That does not mean that in all cells 50% of the ER is devoted to rough and 50% of the ER is devoted to smooth. It just means that whether the cell needs to be whether the cell needs to take on more of a protein production role or a lipid production role will significantly influence the proportion of that cell that is devoted to either one of those ERs. Ribosomes. Ribosomes are the structures that are associated with the rough ER, but the ribosome itself, which are each of these individual bumps, is composed of two subunits. You have a large subunit and you have a small subunit. We're not going to get into the nuts and bolts of protein synthesis right now, but the small subunit for now is the subunit that is going to bind to our mRNA protein, and the large subunit is going to be the site where our tRNAs, which are essentially the molecule that houses the amino acid, and has our anticodon, which is the kind of complementary triplet that is going to bond to our triplet that is associated with the mRNA, which is our codon. Again, I know I'm throwing a lot of details out there. We don't need to know those per se right now, but the large subunit is going to be the site of our tRNA to bind, and our small subunit is going to be our mRNA to bind. But the ribosomes, through the use of these tRNA and mRNAs, are going to carry out protein synthesis. They are 80S within eukaryotes, and they themselves are composed of rRNA, or ribosomal RNA. So it makes sense. Ribosomal RNA constitutes ribosomes. Golgi apparatus is going to look very similar to the ER, but it's very different. For one, the rough ER is an interconnected set of flattened membranes that are associated with ribosomes. The Golgi apparatus is a non-interconnected flattened sac of membranes. Notice that some of them are connected, but this membrane is not connected to this one, which is not connected to this one, which are not connected to these. There is a receiving side and a shipping side of the Golgi. Transport vesicles are going to enter the Golgi from the rough ER and are going to leave the shipping side in secretory vesicles that can either be kept within the cytoplasm or can actually leave through the membrane, which is one way the cell is actually going to communicate with adjacent cells. The cis face is the receiving side. It is the one that faces the rough ER, and the trans side is the shipping side. It is the one that is opposite or on the other side of the Golgi from the rough ER. The rough ER would be up here, and the ribosomes would be attached to it. And the rough ER, utilizing those ribosomes, would produce proteins that they put in these transport vesicles. Vesicles would be received by the Golgi. They would be modified, they'd be repackaged, they'd be shipped out on this trans side, and they would be sent to where they are needed. So the Golgi apparatus is much like a post office. It is the function is to collect, package, modify, and distribute materials throughout the cell. Okay. One thing that I would like for you to kind of pay attention to is that the Golgi apparatus is especially prevalent in glandular cells, meaning the cells that are associated with glands of the body, like the pancreas, because the pancreas, as an organ, the pancreas's job is to secrete stuff. Okay? How does it secrete stuff? It utilizes the Golgi apparatus to secrete to the rest of the body through the membrane, utilizing these secretory vesicles, the products that are produced by the cells of the pancreas. Okay? It's one of the ways that the glandular cells work. Vesicles are these transport vehicles, so to speak. Vesicles are a single membrane that has fluid inside that are used, it's essentially a fluid-filled bubble that are used to transport cellular products. They're very small in size, and all they are used for is just a transport vehicle to transport the cellular products like within those glandular cells. If it is a hormone, or if it is a protein product that was produced by the pancreas cells, or the cells within the pancreas, it's going to produce these products and then package them in vesicles to ship them out to other parts of the body, which is ultimately the purpose of those glandular type organs. Lysosomes are similar to vesicles, but they are very different. Lysosomes are a single membrane that has fluid inside, just like the vesicles, but the protein products that they are containing have hydrolytic or digestive enzymes specifically within them. And so they utilize these digestive or hydrolytic enzymes to break down ingested food, break down unwanted damaged organelles, or even break down the cell if the cell wears out or if the cell dies. That is called apoptosis. And one of the ways that you can differentiate lysosomes from vesicles when you look at actual electron micrographs is that lysosomes are going to always stain really dark. So if you see structures within a cell that look very dark or filled in, they are most likely lysosomes. And that's just because they have a very, very, very high concentration of proteins in the form of those digestive enzymes. And so they always stain very heavily and appear dark because of the sheer number of them within that membrane. Mitochondria are rod-shaped organelles that appear throughout the cytoplasm in a eukaryotic cell. Both plant cells and animal cells are going to contain mitochondria. The unique thing about mitochondria, and we'll talk about it later on in the IB syllabus with the endosymbiotic theory, is that there is two membranes, but the two membranes within the same organelle are vastly different. What I mean by that is the outer membrane, the membrane on the outside, is going to appear very similar to the membrane of the cell itself, specifically the eukaryotic cell itself. This outer membrane is the membrane that regulates, maintains the boundary, so to speak, between inside and outside of the organelle. The inner membrane is very different from the outer membrane. Not only is it folded up into these cristae structures to increase surface area, but the inner membrane actually resembles more of a prokaryotic cell membrane as opposed to the eukaryotic membrane that the outer membrane appears to be. So, this interior or inner membrane is the site of cell respiration and chemiosmosis. It is all about ATP production. That is why it is coiled up or folded up into these structures. It's all about maximizing surface area, decreasing volume. You want to have a lot of room for ATP production inside of that inner membrane. So we've got the outer membrane, inner membrane, and then inside of this inner membrane we actually have a fluid, which is very similar to the cytoplasm within a cell, but it is called a matrix, and it is the site of the Krebs cycle. So in future videos we're going to talk in depth cell respiration processing details, but for now just know the anatomy of the mitochondria itself. We have outer membrane, inner membrane, matrix, and then inside of the matrix, or inside of the inner membrane within the fluid, is actually DNA. One of the biggest kind of differences between the mitochondria and other organelles is that this organelle itself actually contains its own DNA, unlike a lot of the other organelles. An organelle that is very similar in structure to the mitochondria would be the chloroplast, and it also has a double membrane structure, but it too has DNA. One of the cool things about mitochondrial DNA is that not only is it maternally inherited, meaning you inherit the mother's mitochondrial DNA, not your father's mitochondrial DNA, and that's just the result of the egg cell containing the organelles like the mitochondria. The sperm cell does not contain mitochondria. Well, it does to power the sperm cell, but it doesn't have mitochondria in terms of inheritance. But it has maternal DNA, and so it therefore can actually reproduce themselves. So in cells that have high energy demand, actually have a lot of mitochondria per square inch, so to speak. So if there is a high demand placed on energy output or energy consumption within the cell, mitochondria can actually reproduce themselves in order to increase the concentration of mitochondria within the cell itself. So this DNA is circular chromosome. It is very similar to bacteria. And mitochondria are actually found, just like I said, within the majority of cells, but are in much larger concentrations within cells that have high energy demand. Like muscle cells would have tons of mitochondria because it requires a lot of energy to contract and use a muscle like a muscle is actually supposed to be used. Chloroplasts, just like mitochondria, are going to have a double membrane. Notice you have an outer membrane, you have an inner membrane, and then not tied to the inner membrane like the cristae of mitochondria, you have structures within that are perfectly designed to increase surface area to volume ratio. The thylakoids would be these individual flattened discs within the stroma, which is the fluid which is within that inner membrane. And then you have granum. The granum are these stacks of thylakoids. So kind of from outside in, outer membrane, inner membrane. Stroma is the fluid that fills the inner membrane. Thylakoid is the individual flattened disc, and then a stack of these flattened discs would be considered a granum. These chloroplasts only occur in plant cells. These are the sites of photosynthesis. Animal cells aren't associated with chloroplasts, or chloroplasts aren't associated with animal cells because we don't photosynthesize. But like mitochondria, the chloroplasts contain its own DNA. It is in the form of a ring, just like bacteria DNA. It contains its own 70S ribosomes, which is identical to the ribosomes within the mitochondria. They can reproduce on their own, and they both resemble a bacterial cell. You'll notice that the shape of these chloroplasts and the shape of the mitochondria appear to resemble bacteria cells, one of the big pieces of evidence to help support the endosymbiotic theory. They contain numerous flattened sacs called thylakoids. Like I said, the chlorophyll that the plant cell contains is actually contained within the membrane of these thylakoids. It is the light-absorbing pigment, and these thylakoid membranes, as will be addressed in future videos, is the site of the light-dependent reactions, which is the first half of photosynthesis. The fluid within the inner membrane would be the stroma, and it is the site of the light-independent reactions, also known as the Calvin cycle. So, the fluid contains many enzymes and chemicals necessary to complete the process of photosynthesis. The first half of photosynthesis would take place here at the thylakoid membrane. The second half would take place out in the stroma. And so when you put the first half and second half together, you get the full completion of the process of photosynthesis. Vacuoles can be most notably noticed in plant cells. This is a black and white electron micrograph of a plant cell. You can see that this would be a chloroplast, this would be a chloroplast, and then you have this large central vacuole. Notice the central vacuole within a plant cell often takes up the majority of the interior space of a cell, but it is essentially just a single membrane with fluid inside. This fluid can be used to store food, water, waste products. All of these products would be utilized by the cell in certain instances, and so the central vacuole is really just a storage vessel that can be used to store, like I said, food, water, and waste. Plant cells have these large central vacuoles. In animal cells, these vacuoles are often a lot smaller, and that's because animals are typically mobile, meaning we don't have to store as much water in our cells because we have the ability to seek out and find water. Plants are just kind of at the mercy of where they were kind of rooted, and so they have to have the ability to store large volumes of water so that in water scarcity they don't become flaccid, which you can see down here in this diagram. So, cell wall. Last kind of structure we're going to talk about associated with the ultrastructure of cells is that this is kind of an extracellular component, meaning we have now left the membrane and we are now outside of the membrane. When you look at a plant cell, the membrane is right through here. It's associated with the boundary between inside and outside of the cell, but the cell wall is often the only structure seen when you look at a light microscope or a light microscopy, because the cell wall is so much thicker. Okay, so the cell wall is on the outside of the membrane and is actually components, or this extracellular components, that are secreted by plant cells and some fungi and some protists. Okay, but the cell wall consists of cellulose when you are plant, so the plant cell wall is constituted or made up of cellulose. Fungus cell walls, chitin, and bacterial cell walls are peptidoglycan. All three of these substances are used to create a cell wall. And the functions of a cell wall is that you have to kind of create a strong, rigid, hard to digest, and a permeable barrier between outside environment and the outside of the membrane. So it's kind of the first line of defense, but it's often designed to give structural support to the cell from outside of the membrane. And so when you think about it in terms of a plant application, plants are able to achieve long, slender stalks because they have the rigidity of the cell wall. If plants didn't have the cell wall, they would not be able to remain as rigid as they are. So now we're getting kind of into the application portion of the lecture. Now I have a particular plant cell, and I want you to be able to look at these micrographs and identify structures kind of based on what they physically look like. So the first order of business is determining if it's an animal or plant cell, other than the fact that I gave you plant cell in the title. You should be able to identify this as plant cell 1 because it has a large central vacuole, and 2, it has these chloroplast structures. So if we go through this, the plant cell obviously most notably by the vacuole. So this large central vacuole is taking up the majority of the interior space. In fact, it is very, very much larger than the nucleus itself, which is kind of hard to believe in some instances. Mitochondria are really hard to tell sometimes. Sometimes they're very noticeably mitochondria and sometimes they're not. But you have vacuole, mitochondria, and then you have the chloroplast, which we talked about right at the beginning, right? These are the chloroplast structures you can see inside the stacks of grana or the stacks of thylakoid that makes up these grana. Chloroplast tells you that it is a plant cell. You have the cell wall on the outside of the cell membrane, and you have the Golgi apparatus, which again in this particular image is very hard to tell, but Golgi apparatus, one of those eukaryotic organelles, and then you have the nucleus itself, which is associated with the nucleolus and the DNA containing structure. If you think about just kind of what an artistic rendition of these eukaryotic cells would look like, you can see here that there is a difference between the plant cell and the animal cell. There are a lot of similarities. Obviously, both have mitochondria, both have a vacuole, both have a nucleus, both have a Golgi, both have a cell membrane, both have ER, right? All of these structures can be seen in all eukaryotic cells, but now I want to get into kind of trying to deduce a function or draw a conclusion about what these particular micrographs are based on observations. So when we think about kind of making observations, you want to keep structure and function, or structure dictates function, kind of at the core of all of these observations. So what are some of the observations? When you look at this, there is a nucleus present. Okay, we can obviously see the nucleus, and so that right away tells you that it is in fact a eukaryote. So it is not a prokaryote, it is a eukaryote. There is no cell wall on the outside. In fact, there's just a cell membrane. So no cell wall tells you that it is an animal cell. Okay, a lot of ER. I mean, you're probably wondering what all of this kind of weird membranous type stuff is, and that is all endoplasmic reticulum. That must mean that there's high energy demand, because this is all rough ER. Okay, you can see a lot of rough ER associated with this particular cell. There's a lot of mitochondria. Okay, you can see all of these mitochondria, so it must mean that there's a lot of or high energy need. So we've got a high protein demand, high energy demand. A lot of secretory vesicles. Okay, you can not only see a lot of these secretory vesicles, but you can also see a lot of these lysosomes. Okay, so we've got a lot of vesicles, a lot of lysosomes, and so when you put all of these observations together, it tells you that this is likely a cell that specializes in the production and secretion of protein products, right? If you have a nucleus, you have no cell wall, you have a lot of ER, so a lot of protein production and a lot of protein modification, you have a lot of vesicles and a lot of mitochondria, then it really tells you, or you can legitimately draw a conclusion that this cell must specialize in the production and secretion of protein products. Okay, and so what is this cell? This is in fact a mammalian exocrine secretory cell from the pancreas. So if you took a cell from the pancreas of a particular person and you looked at it under the microscope, you would probably see something very similar to this. A cell with high energy demand, high protein product demand, and a lot of secretory vesicles, lysosomes, because that's ultimately the purpose of that particular cell. Another one, this one looks very different because this one has a different function. Okay, so what are some of the particular observations we can make? Again, there is a nucleus present. You can see these nuclei within each of these individual cells. It has to be a eukaryote. No cell wall, has to be an animal cell. Okay, again, this is not a plant, this is an animal cell. The cells right here at the border, okay, and so again, we're talking these cells at this border have really no room. They're very densely packed. Okay, there's not much room between them. They're very, very closely packed, and so it disallows entry of substances. It has to be a really uniform, nicely packed border. Okay, that's something that's very different from the picture we saw before. It has cilia-covered border. Okay, I don't know if you can tell, but all of these along a lot of these structures, okay, all the way along this border are these really tiny projections. Okay, we call that cilia, and cilia have a particular function within cell structure, and so they are typically used to move substances away from the cell. Cilia around the outside of a cell are not typically associated with the movement of that cell. That would be pili and flagella, but they're really designed to move materials around the outside of a cell. So this particular border is probably or probably has something to do with the movement of substances to and from this particular border. Likely to be a protective layer or a layer that benefits from increased surface area to volume ratio, because all of these small microvilli or all of these small projections are going to increase surface area to volume ratio. And so if we draw a conclusion, this is a ciliated epithelial cell from a mammalian lung. So when you look at the interior cells that line the lung, they often look like this. You have to have, a good boundary between them, a really good diffusion path, but also you want to move kind of the aqueous solutions away from those cells so that you can maintain the proper moisture, but also maintain the proper diffusion paths. And this one, okay, this one looks very different. Notice along the outside, you probably see something that is different from the other two previous pictures. We not only have a nucleus present, this nucleus just happens to pretty much dominate this cell, right? So it is a eukaryotic cell. The nucleus is huge. You have cell wall present, so it is a plant cell. But we don't notice any chloroplasts, right? So no chloroplasts, but it's a plant cell. How do you have a plant cell and no chloroplasts? Well, if there are no chloroplasts, there's likely no photosynthetic capabilities, right? So it has to be a cell from a plant from non-photosynthesizing tissue. Right? Not all of the plant, or the whole plant, doesn't photosynthesize. In fact, just the leaf tissue photosynthesizes. There's a lot of other tissues like root tissue, stem tissue, stalk tissue. A lot of those don't have the ability to photosynthesize because, one, they're not in direct access to light, but also they don't have the chloroplasts associated or that are needed to complete those photosynthetic pathways. So this could be a plant tissue or a cell within a tissue that is not directly involved in photosynthesis or just doesn't receive sunlight in general. The vacuoles are relatively small, right? There are no large central vacuoles associated with, so water must always be available, or there has to be maybe not as much water demand on this particular cell, which is different from other cells. So maybe the environmental conditions that this cell is not directly tied to isn't maybe a cell that is associated with direct sunlight. Maybe it's not tied directly to heat. Maybe it's not exposed to wind. Maybe it's not exposed to salinity or pH or whatever, right? The nucleus is relatively large, or the cell size is relatively small comparative to the nucleus. This could mean that this cell is likely to be a new cell that has recently undergone cell division, right? If this cell just divided, or if this was a brand new daughter cell, you would likely think that the nucleus would in fact dominate the cell, and the cell size would be relatively small. Okay, so if we put all of these together, no chloroplasts, no photosynthetic capabilities, really small size, large nucleus, very small vacuoles, means that it is possibly recently divided cell tissue from a plant root. And that, in fact, is the case. It actually is a meristem plant root tip type cell. And so these are actively dividing. The cells don't get very large because they just divide so quickly. Okay? It would make sense that a plant cell associated with a root tip wouldn't contain chloroplasts because it's not in direct access to sunlight, right? It's underground. And it would make sense that it didn't have large central vacuoles because roots are always exposed to water because they're in the ground, right? Totally makes sense. Structure and function, have to be able to look at micrographs, deduce a function from the structures, just make good solid observations and you'll be fine. I think that's it for this video. I'll see you in class. Bye.